So I'm studying for the MCAT, and this is the mind map I made to connect topics like cells, the cell cycle, mitosis, meiosis, and the female and male reproductive systems, as well as the menstrual cycle together. I think a mind map is a great way to study because you can see how different concepts are linked, and in one page you have all of the material that you need to review for the day or for the unit, and it's just better than reviewing 6 pages, which can take a lot of time. So let's get started. In the top of my mind map, also this mind map looks really confusing because there's a lot of information on it, but when you look at it part by part and you see how things are connected, it's actually really easy to understand. And I have pictures as well to help you understand. So if you're studying, let's study together and let's get started. So first, this topic starts with cells. So cells are the main functional unit in your body, right? There are 2 types of cells in your body. There are somatic cells or body cells, and there are germ cells or sex cells. So what are somatic cells? So body cells are any cell in your body, right? Like your brain cell, your heart cell, your muscle cell, your cheek cells. So these cells have 46 chromosomes, okay? And that means that they are diploid because each chromosome has 2 copies. So you have 23 chromosomes plus 23 chromosomes, and the total is 46 because each of the chromosomes has a copy, okay? That's why they're called diploid, and that's why they're called 2N cells, right? You may have seen that. Germ cells, in contrast, are sex cells. So these are your sperm cells and your egg cells. So your sperm cells and egg cells are responsible for fertilization and the progression of life, right? And these are haploid cells, which means that they have only 1 copy of each chromosome, which means that they're N. Okay? If they were 2N, I always used to ask myself, like, what if they were 2N? Well, that would mean that a sperm with 46 chromosomes is combining with an egg with 46 chromosomes to result in a fertilized egg or fertilized cell with 92 chromosomes because 46 plus 46 is 92. And that is not viable with life, okay? What is viable is if you have a sperm with 23 chromosomes that fertilizes an egg with 23 chromosomes, which results in a fertilized cell with 46 chromosomes, okay? That's viable with life. Now that cell can grow and divide and result in the embryo and the progression of life. So that's why these germ cells, they're haploid. So as you may or may not know, these cells, they go through something called a cell cycle. So the cell cycle is essentially the rotation of things that will happen throughout their lives. And remember that these are eukaryotic cells. They replicate via this cycle, okay? So the cell cycle has 4 parts, and they're actually really easy to understand when you look at them this way. The first 3 phases are called interphase altogether, okay? Here, we want to note that the individual chromosomes are not visible because they exist in a less dense or compact form called chromatin. So they look like this, okay? When the cell is about to divide, the chromosomes condense, or the DNA condenses, and it looks like this, these chromosomes, okay? But before we get to mitosis or meiosis, let's talk about what happens in interphase. So interphase consists of G1, S, and G2. G1 is the pre-synthetic gap phase. S is synthesis, and G2 is the post-synthetic gap. As you can see, it kind of makes sense why G1 is called pre-synthetic. It's because it's before synthesis, and G2 is called post-synthetic because it's after synthesis. So what happens in G1? In G1, the cell makes organelles and increases in size, okay? So it's getting ready to divide. It needs to make organelles, and it needs to increase in size. What organelles does it make? It makes mitochondria, endoplasmic reticulum, and such. Okay, so what happens before synthesis? Well, the cell needs to check, do I have everything I need to move on? This is called a restriction point. There are 2 restriction points in the cell cycle, the G1S restriction point and the G2M restriction point, okay? And in this way, the cell is able to check, hey, do I have everything I need to move on? And if I don't, I'm not going to move on. I'm going to stay right here, okay? And this is great because we don't want to divide if there's problems, okay? So after the G1S restriction point, the cell moves on to synthesis. So synthesis is where the cell replicates its DNA. It needs to replicate its DNA so that each daughter cell has identical copies. So remember that during mitosis, the cell goes from one cell to resulting in two daughter cells, right? These are exact copies of this cell. That's how it divides. And for that, it needs another copy of the DNA. So that's why it replicates the DNA. After synthesis, the cell goes through the post-synthetic gap phase. You might have read this in your textbook as being the quality control phase. I always used to wonder, what does that even mean? Well, that means that here the cell is checking to see if it has sufficient organelles for both of its daughter cells. Okay, so again, the cell is checking for quality and of the organelles. Okay, so after G2, there is another restriction point like I talked about, which is called the G2M restriction point. Here, another checkpoint for the cell to check for errors in DNA replication, and also is checking whether it has adequate size and existence of organelles for splitting. Okay, so if this is good, if the cell knows that there's no errors, okay, DNA is fine, it has these organelles for splitting, then it moves on to mitosis, okay, where the cell divides. So one thing I want you to ask yourself is, what if there's a problem, okay? What if there's a problem between any of these restriction points? What happens? Well, the cell goes through something called apoptosis, okay? This is programmed and controlled cell death. You may have heard of something called necrosis. This is not programmed and controlled. Apoptosis, however, is, okay? The cell is saying, hey, I don't have everything I need. There are problems here. Let me unalive myself. Let me go through apoptosis because I don't want to keep dividing. There could be problems if I keep dividing. Something that helps repair damage is P53. However, you may have heard of cancer and how it occurs. This is where the cell cycle control is destroyed, okay? So there's a gene mutation in the gene that produces P53, and that stops P53 from correctly checking the DNA for mutations, and therefore the cell can't stop dividing. It divides and divides with the mutation, and that results in a tumor forming, and that tumor then metastasizes to other parts of the body. It just spreads, okay? And that is what cancer is essentially. So as you can see, the cell is really smart in a way, right? Because it knows what to check and when to check so that all of these phases are complete, and they're complete in the right way, and we can move on to dividing, okay? But if there's issues, obviously the cell has ways in which it deals with that. So now that we talked about cells and the cell cycle, let's talk about what happens during the M phase, okay? Well, one thing I also want to note is here we also see that there's a G0 phase. I didn't talk about that, but basically when the cell isn't dividing, you might be asking, what is it doing? Well, it's spending time in the G0 phase where it's simply just carrying out its life processes, okay? And you can use this visual to look at how the phases of the cell cycle, and also these notes you can look back on. So as we talked about, we go from G1, S, G2, and the M phase. The M phase is where cell division happens. So back to talking about cell division. So the somatic cells, your body's cells, they essentially divide through something called mitosis. Let's talk about that first, okay? So mitosis essentially has 4 different phases. As you can see here, we just talked about interphase. So during interphase, like I told you, the DNA is in its less dense form. It's called chromatin. It looks like a bunch of spaghetti, right? And this is what it looks like during interphase. However, during prophase, the chromatids condense, the nuclear membrane disappears, and the central pairs form. So, in a simple way to understand that, what is going on, right? Well, the cell is getting ready to divide. This DNA is hard to divide because it's so complex, convoluted, right? We want to get it in its condensed form, so it's easier to see and it's easier to divide, okay, in half. Because at the end of mitosis, we want to result in 2 daughter cells, okay? We want to take this one cell and end up with these 2 cells that are exactly the same, okay? And in order to do that, we need to make sure that the DNA is split properly. And in order to do that, we need to make sure that the DNA is in its condensed form. So that's essentially what's going on in prophase. The DNA is in its condensed form. These are called chromosomes, okay? This is a sister chromatid. This is a sister chromatid. Together, they are chromosome. This is a chromosome. And in the center, it's held together by something called a centromere, okay? I think I drew that up here. Okay, so as you can see right here, this is a centromere. And then these are the sister chromatids, okay? It's basically double-stranded DNA. This is what double-stranded DNA looks like, okay? So as you can see here, the nuclear membrane also disappears. So this nuclear membrane here, it's disappearing here because the cell is going to start dividing, right? So essentially, I want you to pay attention to one thing. So in the beginning, the cell, it has 2 copies of DNA, and they're both double-stranded, okay? By the end, it's going to be 2 copies of DNA as well, except it's going to be single-stranded. That's what mitosis results in. But how does that exactly happen? So once the DNA is in its condensed form and the essential pairs are formed and everything, we move on to metaphase. Metaphase is really simple to understand. Meta, I believe, means middle. So the chromosomes essentially meet at the middle of the cell. This is called the metaphase plate. And the essential pairs that formed over here, they move to opposite ends of the cell, okay? And they use spindle fibers to attach these centromeres, okay, of these chromosomes. After that, during anaphase, the sister chromatids, they're pulled apart. So these sister chromatids, they're pulled apart. And as you can imagine, the cell is going to start splitting here, right? Because now we've split the DNA. So during telophase, a new nuclear membrane will form, the chromosomes will uncoil, and the spindle apparatus will dissociate, okay? Because division is complete, okay? And remember that cytokinesis also happens where the cell actually splits, okay? Right here. So as you can see, this is how our cells, our body cells, go from one cell to dividing into 2 cells with the same DNA. And when is this relevant, you might be asking yourself? Well, think about this. When you were a kid and you learned how to ride a bicycle, or you were running around with your friends in the playground and you fell and you scraped your knee, how did that wound heal? Okay, how did you grow taller? How did your hair grow longer? Well, your cells are dividing, okay, and those are your body cells. So like your skin cells have to divide to cover up that wound, right? Well, how do they do that? Well, they use mitosis. Okay, so they take the cell that's already there and they divide it into identical cells. These are called daughter cells, where the DNA has 2 copies, except it's single-stranded. Okay, we started with double-stranded DNA. We end up with single-stranded DNA. And that's how mitosis works. Okay, so how does meiosis work? Okay, meiosis is the process of cell division, I wrote over here, that reduces the chromosome number by 1/2 and results in haploid gametes. So here, how do our sperm and egg cells divide? Okay, how does that happen? Well, meiosis has 2 phases, meiosis 1 and meiosis 2. Let's start talking about meiosis 1. Meiosis 1 essentially is a separation of homologous chromosomes. As you can see in the beginning of meiosis 1, our cell looks similar to how it did during mitosis, okay? So we have a cell with homologous chromosomes, okay? So essentially we're starting with double-stranded DNA and 2 copies of that DNA, okay? So 1 copy and a second copy. And it's double-stranded. 1, 2, right? So prophase 1, nothing different really happens from mitosis. Essentially, same things happen. The chromatids condense, the nuclear membrane disappears, central pairs form, right? Except something different that's really, really important happens. It's called crossing over, okay? And this sort of answers why we look different from our parents, right? You don't look exactly like your mom. You don't look exactly like your dad. Your parents don't look exactly like their parents, right? You look like a mix of your parents, okay? And that happens through crossing over. So essentially homologous chromosomes are in the cell like this, and parts of them are touching, okay? And then parts of them exchange. And that's how we result in genetic diversity. Okay, that happens during prophase. After that happens, the chromosomes, homologous chromosomes, essentially line up and they divide. And this division, they're pulled apart, and this is called disjunction. You can use parts of the words, like the prefix and the suffix, to understand what that word means. So if you don't know about what disjunction means, well, this means apart, and junction is the point where 2 things are joined. So we're pulling those 2 things apart. okay? You may have heard of something called non-disjunction. That's a problem that occurs. Non means not, okay? So there's some problem and the homologous chromosomes do not get pulled apart and you have an extra chromosome in a cell, things like that. So that's what non-disjunction is. All right. So after anaphase 1, when the homologous chromosomes are pulled apart successfully, we move on to something called telophase. Essentially, you have 2 cells now with 1 copy of DNA in each cell, and that copy of DNA is double-stranded, as you can see. And that's when we move on to meiosis 2. So the main thing that you should know here is that meiosis 2 is a separation of sister chromatids. So we went from these cells that split from here, right? And they essentially further split. And what happens is through metaphase, the chromosome in this respective cell, it lines up in the middle, and then during anaphase, the sister chromatids, now we're talking about the individual strands, they get pulled apart. And at the end, we result in 4 cells. Okay, and these are 4 haploid gametes. Essentially, these are haploid cells, and they're single-stranded. So we reduce the chromosome number by 1/2. Okay, so instead of having diploid cells, we have haploid cells with only 1 copy of DNA. That's what haploid means, and it's single-stranded. And these are what your germ cells actually look like. Okay, so they have 1 copy of each. Okay, and essentially that is how meiosis and mitosis are similar and different, and how the somatic and germ cells use mitosis and meiosis to divide. One thing I want you to know is that something that helps in this is cyclins and cyclin-dependent kinases. So cyclin-dependent kinases, they're only active in the presence of the right cyclins. And cyclins are things that when they increase and decrease in various parts of the cell cycle, they allow certain things in the cell cycle to happen. So cyclins actually bind to the cyclin-dependent kinases, and they make activated cyclin and CDK complexes. And essentially, these complexes then phosphorylate certain transcription factors to allow phases of the cell cycle to occur. So this is just something to note. These help in phases of the cell cycle and are something that you should know about. So now we talked about cells and how they divide. Let's talk about the female and male reproductive systems and the menstrual cycle and how all of this comes together. So first, let's talk about male reproductive anatomy and physiology. So 4 things to note here. The first thing to note is that the main gene that codes for male gonad formation, so male gonads are like male sex organs, is the SRY gene. Okay, something you should just know. Second thing to note here is that the testes are the functional unit of the male reproductive system, okay? And this is where most of the testosterone is produced and where sperm are produced, okay? Remember that sperm then go on to fertilize egg, right? So how do they actually get out of the body and move toward the egg? Well, this is the process. So a lot of people use this acronym. It's called 7-UP. So the sperm essentially gets produced in the testes, right? And then it's essentially in the seminiferous tubules. So the seminiferous tubules are in the testes, and that's where sperm is produced. And from there, the sperm, they move to the epididymis. This is where they are matured and where they are stored. The epididymis sits on top of the testes. So the sperm are first in the testes, specifically in the seminiferous tubules, like we just talked about. Then it moves to the epididymis. Then it moves to the vas deferens. Then the ejaculatory duct. And from there, the urethra and out through the penis. Okay? When it moves out, the sperm is actually mixed with seminal fluid. And seminal fluid is produced by these 3 things: the Cowper's gland, seminal vesicles, and the prostate. That's what seminal fluid is made of. And one thing also to note is that before the sperm enter the urethra, the seminal vesicles also clean out the urethra. Because, as you know, in the urethra, there's urine. And urine is made up of urea. And urea is acidic. So it will lower the chance of fertilization because it will kill the sperm, okay? So you want as many sperm as you can get because you want fertilization to be successful, right? You want to aid in the progression of life. And for that, you need to clean out the urethra before the sperm enter. And that's what seminal vesicles are responsible for. Just something to note. And here this is a diagram I drew to better understand the process of sperm getting out of the male body. So essentially we start in the testes, right? Right here, we move to the epididymis. Then we are in the vas deferens right here. And then we go to the ejaculatory duct. And then we exit out through the urethra and the penis right here. This is something diagram you can use to help yourself see how the sperm move out. Something cool I want you to note is you may have noticed that the testes hang outside of the body. Okay, they're in the scrotum, which drops and elevates. And they hang outside because the body temperature is too high for the sperm survival. Okay, that's why they drop and elevate. The scrotum drops and elevates via muscles, depending on the temperature of the body. And this is how the body is thermoregulating ideal sperm temperature or ideal body temperature for the sperm formation. Okay, so there are two ways in which the body is making sure that the sperm survive, and we have as many sperm as we can get in order to essentially shoot our shot and fertilize that egg. So two ways. One way, the seminal vesicles are cleaning out the urethra, making sure there's no urea. Another way, the testes are outside the body in the scrotum, and the scrotum drops and elevates depending on the body temperature to make sure that the sperm are good. It's kind of cool. So you might be asking, well, how are these sperms even forming? How is that happening, okay? And this is called spermatogenesis, which is basically sperm maturation. It's regulated by a hormone called FSH, follicle stimulating hormone, okay? Where you start with spermatogonia, which are diploid sperm, and then they replicate and you have primary spermatocytes. After the first meiotic division, remember we talked about meiosis, we end up with secondary spermatocytes. And then after the second meiotic division, so these cells, they go through another meiotic division, and we end up with spermatids. And then when these spermatids, they mature, they result in these spermatozoa. So during maturation, they develop these tails, right? And then we end up with 4 sperm. Okay, so this process happens over and over with these diploid sperm cells, and they develop into these spermatozoa, which are ready to fertilize that egg, okay? So we talked about meiosis, so you can kind of imagine how these diploid cells go to become these haploid cells, right? Haploid gametes. And that's why that's relevant. Now let's talk about the female reproductive system. So the sperm, it comes in, right, to fertilize the egg. But what is the egg and where is that? Right? And what parts of the female reproductive system should you know for the MCAT? Well, the female reproductive anatomy includes the fallopian tubes, the ovaries, and the uterus, okay? So these are the fallopian tubes. They are essentially the site of fertilization, okay? And these are the ovaries right here. They are the functional unit of the female reproductive system. And as we talked about here, the testes are the functional unit of the male reproductive system. And you might know this or you might not know this, but this is kind of cool. The testes and the ovaries, they're derived from the same embryonic structures, okay? So these are both the functional units, and in the embryo, they are derived from the same structures, okay? So essentially, the ovaries are the functional unit of the female reproductive system, and they have thousands of follicles. And one follicle each month, so a follicle kind of looks like this, it develops into the egg, okay? And that egg then travels to the fallopian tube where it is fertilized, okay? And then it travels to the uterus, and that's where it implants, okay? And then it grows and grows and grows. And then when the mom gives birth, it's through the vaginal canal, right? If it's a normal delivery. So how does the female gametes, the eggs, actually produced? We talked about spermatogenesis, sperm maturation, but how are female gametes produced? Well, we start here, as you can see, with a germ cell. Okay? And we talked about germ cells and what they are. And essentially, that germ cell, it develops into or divides into a secondary oocyte. Before that, it starts as an oogonium. It's a diploid cell. Okay, so this germ cell develops into a diploid cell. And here, this diploid cell becomes a primary oocyte. Okay, because now the cell is getting signals to develop into an ovum, right? From there, puberty hits. So all of this happens before puberty, okay? So before puberty, we have a primary oocyte, the baby girl, right? But after puberty, the girls' primary oocytes, they develop into secondary oocytes through meiosis. Here, meiosis had started, but it paused because puberty had not hit yet. But after puberty, this primary oocyte, it essentially divides into a secondary oocyte and a polar body. Okay? During this time, it's arrested in metaphase. But later on, it develops into an ovum. This ovum, it essentially gets fertilized by the sperm. This ovum is what gets fertilized. And after fertilization, if it's successful, we result in a fertilized egg. We'll talk more about this in the next video about fertilization and embryogenesis, but essentially this is how female gametes are produced. We go from a germ cell, which develops and divides into a ovum, which is then fertilized. Okay, so let's review. So before birth, the baby girl has primordial germ cells, and they migrate to developing ovaries. Okay, the germ cells are called oogonia, and they undergo mitotic divisions. Some differentiate into primary oocytes during fetal development. So some of the cells, they differentiate into these primary oocytes. And then meiosis 1 starts, but arrests in prophase until puberty. And then, like I said, during puberty, some of the primary oocytes, under the influence of follicle-stimulating hormone, which is an important hormone in the body, these primary oocytes, they resume meiosis, and it results in 2 daughter cells. One secondary oocyte, and then another polar body. Essentially, this is unequal division. Okay, this cell, something I want you to pay attention to is this cell, when it divides through meiosis, it results in unequal division. So this cell, this secondary oocyte, has more cytoplasm than this cell. That's why this polar body forms, okay? So this secondary oocyte is actually what gets excreted during your period, during menstruation. Okay, and then when it's excreted right here, right, so it's sent from here to here, this is when it has the potential to be fertilized by the sperm, okay? And essentially, this polar body, it degenerates, okay? So this is an example, again, of how meiosis produces the female gametes. We talked about sperm maturation and production, but now we talked about female gamete maturation and production as well. And as you can see here, we use our knowledge of meiosis to see how this can happen, but also we note that here, instead of meiosis resulting in equal division, we resulted in unequal division, which resulted in the polar body and the secondary oocyte. So now you know about these male and female reproductive systems and how these gametes are formed, okay? Now let's talk about how, what happens during menstruation. What happens when the secondary oocyte gets excreted during menstruation? Well, that is the menstrual cycle. The menstrual cycle, this is an easy way to see how it works. It's not really drawn in a circle, which is sad, but hopefully this is easier for you to follow. Okay, so let's start with the follicular phase. The menstrual cycle has 4 phases, about 4 phases, right? So the first phase is the follicular phase. So essentially, the female body has a hormone called GnRH. It's called gonadotropin releasing hormone, okay? This increases. This increase leads to the increase of follicle stimulating hormone and luteinizing hormone, which leads to estrogen secretion. The ovary actually produces this estrogen, and when it gets secreted, it puts a negative feedback on this GnRH hormone, okay, right here. However, it also leads to the thickening of the endometrium, okay? So in the beginning, we had a high level of GnRH, which led to a high level of FSH and LH, which led to estrogen being secreted. However, this estrogen turned off this increase and instead allowed the thickening of the endometrium, okay? Remember that this is where that egg will be taken care of, right? You may have also heard this of being the decidua. So this is where the deciduas, essentially blood vessels are forming, mucus is being produced inside of the endometrium. Okay, this is what's happening here during the follicular phase. During ovulation, this is still high, right? FSH is still floating around, so estrogen levels are still increasing and increasing, but then they eventually peak, and that leads to a feedback flip. So since estrogen has now peaked, okay, highest it can go, GnRH is back to being secreted. So it's increasing, which again, as we can see from the pattern here, leads to an increase in FSH, follicle stimulating hormone, and luteinizing hormone. Remember, hormones are what regulate your body, help things occur in your body. So after GnRH increases, FSH and LH increase, okay? And this leads to the LH spike. That's important. But let's talk about first what happens here. So these two increase, which increases in estrogen. See, look at the pattern here. FSH and LH increase. It leads to estrogen secretion. So again, estrogen is increasing. This causes ovulation. Okay, ovulation is when the egg, the secondary oocyte we talked about here, it breaks out of the ruptured follicle and has a potential now to get fertilized. Okay, so it breaking out is actually what results in that the pain you get in your abdomen during your period. So after the egg has broken out, we go on to the luteal phase. So remember we talked about the LH spike? Well, LH spike actually results in the ruptured follicle to becoming the corpus luteum, okay? That's what results in the corpus luteum, and this releases progesterone, okay, which maintains the lining, okay? Progesterone maintains, progesterone protects. You can think of it like that. The uterine lining. Remember, the uterine lining, if it results in, if pregnancy happens, if the egg implants, then it does not shed. But if there's no implantation, if there's no fertilization, there's no implantation of the egg, we result in our period. And the period is actually the shedding of that uterine lining, okay? But one thing I need you to know is that during the luteal phase, after the formation of the corpus luteum and the release of progesterone, the GnRH, FSH, and LH all go down because we want to prevent the ovulation of multiple eggs, which is so cool, all right? We don't want this to happen. The body's like, okay, we're good to go. So that's why these three things, they lower, okay? So if there is implantation, it results in pregnancy, okay? We'll talk about implantation, we'll talk about fertilization, and we'll talk about the growth of the embryo in the next video. But essentially during implantation, the embryo, it secretes this BHCG, or beta human chorionic gonadotropin, which is an analog of LH, but the corpus luteum maintains progesterone concentration secreted by LH until implantation and placenta formation. So we noted here that LH is what actually stimulates the ruptured follicle to become the corpus luteum. So now we're asking ourselves, well, if the embryo secretes beta HCG, which is an analog of LH, doesn't the corpus luteum, like, what happens? Like, isn't it that bad? Well, no. The corpus luteum still maintains its progesterone concentration to protect that lining, to maintain that lining, okay, because this progesterone is secreted by LH until implantation and then placenta formation. And the placenta essentially takes over the secretion of estrogen and progesterone, which inhibit GnRH secretion, okay, for this reason. okay? So if there's no implantation, what happens? Well, menses, your period, your menstruation cycle, that's what happens. The corpus luteum essentially becomes insensitive to LH, okay? Which leads to the decrease of progesterone and estrogen and the uterine lining shedding. Reminder that these are what maintain and thicken the endometrium normally, okay? But since we don't need that, no baby, right? The uterine lining sheds, okay? Essentially, what happens after this? Well, the shedding of the uterine lining occurs because of the drop in progesterone, which we talked about from the previous cycle, and that takes off, eventually it takes off the negative feedback of the GnRH, okay? So instead of GnRH secretion being inhibited because of pregnancy, well, here, the shedding of the uterine lining leads to the negative feedback of the GnRH being taken off, and then eventually we have this whole thing happening again, okay? Because GnRH can now increase, which leads to an increase in FSH and LH, which leads to estrogen secretion, which leads to thickening of the endometrium, okay? Eventually ovulation and the egg breaking out of the ruptured follicle, and then back to luteal phase, okay? This is how you can look at the menstrual cycle. So what happens during menopause? Your question might be, well, as you know, during menopause, a woman ages and her ovaries become less sensitive to FSH and LH, which leads to atrophy. What is atrophy? Well, it's when new follicles that are made, they gradually die off, okay? Which leads to a decrease in the estrogen and progesterone. But why? Well, because these follicles, they are the main source of these hormones. So when they die off, these lessen, these decrease. And menstruation stops, okay? We still have an increase in FSH and LH, but with minimal response, okay? So these hormones, they maintain ovarian tissue, and since the hormones decrease, the ovary shrinks and becomes fibrotic, or tough and scar-like. So this is essentially what happens during menopause. Okay, so now let's review the two hormones that I keep talking about, FSH and LH. Okay, so in women, FSH is responsible for estrogen production, right? Or secretion, really. In men, it's responsible for spermatogenesis. We talked about spermatogenesis. Remember, that's sperm maturation. So it's actually influenced by this hormone, FSH. In women, LH, luteinizing hormone, results in progesterone, okay? But in men, it results in testosterone and also secondary male sex characteristics like facial hair. And both of these hormones are regulated by GnRH and gonadotropins, okay? So GnRH is gonadotropin releasing hormones, okay? So now you know about the cell cycle, how sperm and egg are produced, and how they play a role in sort of fertilization, how they're formed, and how the menstrual cycle works, which is important to know for the next video. Hopefully you found this helpful. Hopefully you saw how these topics sort of connect to each other. This is one page of notes, only one page for all of this information. And hopefully it's helpful for you. Good luck studying.